Hi everyone, my name is Ceren Tozlu. I'm an instructor in the Department of Radiology at Weill Cornell Medicine and a visiting fellow in the Department of Computational Biology at Cornell University. Today we will explore how statistical approaches can be used to better understand the complex relationship between brain function and behavior. I will start by discussing one of the most commonly studied forms of brain connectivity, which is functional connectivity. Functional connectivity can be measured using neuroimaging techniques like function MRI, EEG, and MEG, which allows us to observe spontaneous functional activity in the brain. And in this video, we can see functional activity captured by fMRI, where yellow areas indicate regions with synchronized functional activity. So how we can measure functional connectivity? There are various neuroimaging techniques that we can use to measure functional connectivity, but I will present functional MRI here. Functional MRI or fMRI allows us to measure blood oxygen level dependent time series, or we can call them BOL time series, on each voxels in the brain. And voxel means three-dimensional pixels. And on this figure, x-axis represents the repetition time or sampling rate, and y-axis represents the amplitude of the fMRI time series. On this figure, we can see fMRI time series measured on three different voxels represented with three different colors. And with the help of a brain atlas that divides the brain into different brain regions, we can calculate the regional fMRI time series. And functional connectivity is computed as the correlation of the fMRI time series measured on each pairs of brain regions. For example, here there are regions N and P. The functional connectivity between these two regions are the correlation of the fMRI time series measured on these two distinct brain regions. We can show functional connections in different forms, either using a brain plot or using a mathematical form, meaning it as a matrix. On this brain plot, black dots represent the brain regions, and the edges represent the functional connections, and darker and thicker edges represent stronger functional connections. And if we divide the brain into 86 brain regions, we can represent functional connectivity as an 86 to 86 matrix, where the upper triangle of the matrix is identical to the lower triangle of the matrix, and here each cell represents the correlation of the fMRI time series measured on region N and the region P. And the diagonal of this matrix is identical to 1. So when we use functional connectivity in a statistical approach, or if you want to explore this functional connectivity, we can either use upper triangle or lower triangle of the matrix after we remove the diagonal. And in this slide, I will explain how we can use function MRI and machine learning techniques to better understand brain and behavior mapping. We can use different neuroimaging techniques, including MRI, to measure functional activity in the brain. For example, as I mentioned before, we can use fMRI to measure BOL time series for each subject separately. For example, on this figure, this BOL time series can be measured on each brain region. Then we can create functional connectivity matrices, where lower triangle and upper triangle of the matrix is identical. And when we want to use functional connectomes in a statistical modeling, we can vectorize this functional connectome by only using upper triangle or lower triangle of the matrix. Here I only represent, for example, the upper triangle of the matrix. And if we have an 86 to 86 matrix, and if we use only upper triangle of the matrix, we can create a vectorized functional connectome with a dimension of 3655. After we obtain this vectorized functional connectomes for each subject, what we can do, we can concatenate all these vectorized functional connectomes for all subjects that we can use later in a statistical modeling, for example, machine learning technique. This machine learning and functional connectomes combined together can be used for different purposes, including diagnosis purposes, to distinguish patients from healthy individuals, or to predict disability level, for example, to predict memory loss in people with Alzheimer's disease. So there are different ways of exploring this functional connectivity using machine learning techniques, including diagnosis purposes, prognosis purposes, or treatment selection or development. Or we can use in-basing science to better understand the brain and behavior relationship, or to better understand the neurobiological mechanisms underlying the disease pathology. As the functional connectivity vectors are high-dimensional, because even when we use a brain atlas containing 86 brain regions, we end up having a vector with a dimension of 3655. This is a high-dimensional data that include complex relationship within the data that can be explored with machine learning techniques. And machine learning techniques generally exist on a spectrum between interpretability and accuracy. On one end of the spectrum, we have simple models like linear regression and decision trees, and these models are highly interpretable, meaning we can easily understand and explain how they make the predictions. However, they may sacrifice some degree of accuracy when dealing with complex nonlinear relationships in the data. On the other end of the spectrum, we have more complex models like deep neural networks and ensemble methods such as random forest and gradient boosting. And these methods tend to be more accurate because they can capture more complex interactions within the data. However, their complexity often makes them less interpretable, meaning it is harder to understand why they make these certain predictions. So choosing the right model using interpretability and performance is important, and it involves balancing this trade-off between interpretability and accuracy. And in some cases where interpretability might be more important, like healthcare, where understanding the decision-making process is crucial. On the other figure here, we can see how different pipelines are impacting the prediction performance. So here we are seeing visualization of a possible combinations of options in the pipeline to predict fluid intelligence from functional connectivity in two different data sets. One is from Human Connectome Project, and the other one is UK Biobank. And the lines are color-coded according to their prediction performance. So lighter colors represent higher prediction accuracy. As you can see on the figure, the linear regression with elastic net regularization technique outperforms more complex deep learning techniques such as conventional neural networks. Therefore, I will introduce the linear regression models in this slide as they are more interpretable. Linear regression is a statistical method to model the relationship between a dependent variable and one or more independent variables. For example, on this equation that represents a simple linear regression, y represents the dependent variable that we are trying to predict, for example, cognition, and x represents the independent variable that is used for prediction, for example, age. And beta 0 represents the intercept. It means that it is the value of y when x is 0, for example, the cognition of a newborn baby when the age is 0. And beta 1 is representing the slope of the line, as you can see here. And slope is the association between x and y. And it is representing how much y is changing when we increase x 1 unit. For example, how much cognition is changing when we increase age 1 year. However, in the case of neuroimaging data, we have too many variables that we need to handle and we need to combine to better understand cognition. For example, in functional connectivity metrics, when we vectorize them, we have 3655 features that we need to combine all together to predict cognition. So we need to use, in this case, multivariable linear regression models that we can better understand the complex relationship between neuroimaging data and cognition. Additionally, we can also add age and sex and brain volume to take into account the confounding factors of these features. However, when we have a large number of predictors that are highly correlated with each other, it can cause instability in the estimates of the regression coefficients. For example, in our case, when we use neuroimaging data, we are dividing the brain into 86 brain regions, for example, and these brain regions are not independent from each other. They are highly depending on each other. So the neuroimaging data that we extract from these brain regions are highly correlated to each other. And when we have highly correlated data, it can create instability in the estimates of the regression coefficients, and this instability can lead inflated variances, making the model more unreliable and less generalizable to a new data. Therefore, to address this issue, we can use a technique called regularization technique or any other penalization technique like lasso or elastic net, but I will present regularization technique here. This technique helps by adding a penalty to the size of the regression coefficients, and this penalty shrinks the coefficients which stabilize the estimates and reduces the variance. For example, the formula here represented here is the function for ridge regression, and for the ridge regression we are trying to minimize this overall equation, and we try to minimize the sum of the squared errors between the actual output here yi and the predicted output here yi hat plus a penalty term, and this penalty term is the sum of the squares of the coefficients. So these are the coefficients that I mentioned, the slopes that I mentioned in the previous slide. And we are trying to also minimize this part as well. And this part is scaled by a regularization parameter called lambda. The larger value of the lambda, the greater penalty applied to the coefficients, and forcing them to shrink towards zero. So higher the lambda is, forcing means that we are forcing the beta parameters towards zero. And this shrinking helps to prevent overfitting by discouraging overly complex models. And finally, when we increase lambda, the penalty on the coefficients becomes stronger, and this trade-off allows us to control the complexity of the model. So in machine learning techniques, we need to optimize this lambda by trading off between complexity of the model and instability of the coefficients. By selecting the appropriate value for lambda, we can achieve a balance between the model accuracy and generalizability, leading to more robust predictions. In any machine learning model, one of our key interests is to understand the impact or contribution of each predictor on the dependent variable that we can measure via beta estimates in a ridge regression. For example, we would like to understand the contribution of age in predicting cognition. However, unlike ordinary linear regression, ridge regression introduces a twist by applying a penalty to the coefficients, effectively shrinking them towards zero, as I mentioned in the previous slide in the equation. While this penalization is what makes ridge regression robust, especially in cases where multicollinearity is a concern, like neuroimaging data, it also creates what is known as a bias-variance trade-off. On one hand, it decreases the variance, leading more stable estimates of the variable importance. But on the other hand, it increases the bias because the prediction is not purely based on the data's fit, but influenced by the regularization parameter. So how we do overcome this bias introduced by this regularization? How we can better interpret the feature weights obtained with machine learning techniques? One approach is to look at the relative importance of the predictors rather than looking at the feature weights and try to interpret them. After regularization, the order of the importance among variables is determined by the size of the coefficients. So what we can do, we can order the coefficients based on their amplitude in absolute terms. So rather than interpreting the coefficients, we can interpret their order. Another technique that we can use to increase the interpretability of the estimate is the half transformation of the beta estimates. And this involves multiplying the weight vector, it means that the beta estimates from ridge, by the covariance matrix of the input data sets and the inverse covariance matrix of the output data sets. And this transformation provides us with a more accurate representation of true importance of each predictor, adjusting with the covariance of the input and the output. When we apply machine learning, cross-validation is a crucial technique used in machine learning to assess how well a model generalizes on an unseen data sets. So instead of relying on a single training and test split, cross-validation provides a more robust estimate of a model performance by using multiple splits of data. So here we are representing a nested cross-validation where we are dividing data into 5 folds. So there are different ways of dividing data. One of them is k-fold cross-validation, that we are dividing the data into k-folds. And another one is that we can use leave one out cross-validation, that the test data represent one sample and we train the model using the remaining samples. Here on this figure, I'm presenting 5-fold cross-validation, where the original dataset is divided into training and dataset using 5 folds, and we are using 4 folds to train the model and 1 fold to test the model. And again, we are dividing the training datasets into 5 folds because we are using nested cross-validation. And when we divide the training data into 5 folds, we are using 4 of them to test a hyperparameter, for example, lambda in ridge regression, and we are using 1 fold to test this hyperparameter. And we repeat this algorithm for different versions of hyperparameters and for different versions of folds. And this inner loop helps us to find the best hyperparameter. For example, in the case of ridge regression, we are trying to find the best lambda. After we find the best lambda using the training dataset, we fit our model using training dataset and the best hyperparameter, and test our model using the test datasets to identify how well we can predict what we are trying to predict. It is important to note that we need to repeat this inner loop and outer loop multiple times. For example, I usually repeat this inner loop 5 times and the outer loop 100 times. So the way that I am dividing my data, my original data, into training and test data sets are not impacting the results. So we can use different combinations of faults when we are trying to make a prediction. So we can have more robust and generalizable results. Measuring prediction performance is essential to determine how well our model is performing on a new unseen data. And here I will present the prediction performance metrics for regression problems. And there are different ways of measuring the prediction performance, and I will present the most frequently used performance metrics for regression problems, including correlation and mean squared error. There are two kinds of correlation metrics. One of is Pearson correlation coefficient and the other one is Spearman correlation coefficient. Pearson correlation coefficient measures the linear relationship between the predicted values and the actual values, and it ranges between +1 and -1, where +1 means a perfect positive linear relationship and -1 indicates a perfect negative linear relationship, and 0 indicates no linear relationship. This metric is most useful when a relationship is between the variables are expected to be linear. And when the relationship is nonlinear, we usually use Spearman correlation, where we measure the strength and direction of the monotonic relationship between these two variables. And Spearman correlation is also ranged between +1 and -1, but unlike Pearson's, it doesn't assume a linear relationship between these two features. And Spearman correlation is particularly useful when dealing with nonlinear relationships or when the data contains outliers that might distort Pearson correlation. And second metric we usually also use is mean squared error, which is a common metric that is used to measure the average squared difference between the actual value and the predicted value, which are representing here. And the error is presented here. Y is the actual value, for example, the actual cognition score, and here the line represents the predicted outcome. And when we are having a vertical line from the actual value, we can find the residual error. And MSC is measured as the average, means here average squared difference between the actual output and the predicted output. And lower mean squared error value indicates a better model performance, as it shows the predicted value is close to the actual value. And MSC is particularly sensitive to the outliers because it squares the error here and giving more weight to larger errors. When applying a machine learning technique for brain and behavior relationships, there are multiple areas that we need to be more careful. And one of them is the imbalance in the covariates. It is crucial to ensure the covariates like age and sex and other demographic features are consistently represented across training, test, and validation data sets. For example, if 60% of our data is male and 60% of our data is female, we want to maintain this ratio across all data sets to avoid skewed results. For example, we don't want to train our model in males only and test our model only in females. So we need to keep the same ratio in the training, test, and validation data sets. Next problem is the class imbalance, which is a common issue, especially in classification problems where one class, like cases like Alzheimer's disease, is much smaller than other like controls. For example, if we have an classification problem that we are trying to distinguish people with Alzheimer's disease compared to controls, we can have more healthy individuals than people with Alzheimer's disease. And if we have more cases or more subjects from one class than other class, this imbalance can cause the model or machine learning technique to favor to majority class, leading to poor predictive performance to minority class. So our model can tend to better classify the majority class than the minority class, so we can have poor prediction performance for the minority class. So to overcome this problem, we can use either oversampling or undersampling technique in the training data set to balance these classes. But undersampling is not usually preferred technique, as we need to remove the subjects in our data set, or oversampling is like copy pasting the same subjects may not be preferred. There is also a technique called SMOTE, a synthetic minority oversampling technique, which creates synthetic examples of minority class by looking at the distribution of the data, and this is a very frequently used technique to overcome the class imbalance in the classification studies. And another thing that we need to be more careful is to use balanced accuracy instead of accuracy metric, because balanced accuracy accounts for class imbalance by considering both sensitivity and specificity rather than only relying on simple accuracy, which might be misleading. Overfitting can also occur when the model fits well to the training dataset, and this problem can lead to poor generalization on a naive test datasets. And to prevent this problem, we can use regularization technique, like I mentioned, ridge regression or elastic net or lasso techniques, and we can apply this to penalize overly complex models and help to maintain a balance between bias and the variance. Finally, the site effects refer to the variability that can arise from different data collection sites or protocols. This is especially relevant in multiple site studies where different scanners or protocols or sample characteristics can introduce unwanted variability. For example, in one site, there might be more older people than in another site, or in one site there might be more female subjects than other sites. So to overcome the issue about scanner or protocol differences, data harmonization might be needed, and there are different techniques, but the most frequently used technique is combat to harmonize the neuroimaging data. However, this technique doesn't take into account the different sample characteristics such as age and sex distribution, which might occur in two different sites. That's why there are different naive approaches like combat LS or other techniques as well to take into account different age and sex distribution in different sites. And we can also incorporate site as a coverage in our model to account for sites related variability. It means that we can, in addition to adding neuroimaging data, age, sex, and brain volume, we can also add the site as a coverage in our model. And in some cases, site certification might be also needed, where the model is trained and validated separately on data from each site. For example, we don't want to train our model using one site only and test our data set in another site. So, because in this case, our model can overfit to one site and can provide a poor prediction performance on other sites. So, site certification might be needed when we are applying a cross-validation. It's also important to validate our results using the data from each site independently to ensure that the model generalizes well across different conditions. So we want to make sure that our model doesn't only perform well in one site but all sites. In this part of the presentation, I will show you how to load the fMRI time series, convert these time series as functional connectivity matrices as well as functional connectivity node strength. We will then use the functional connectivity node strength to predict total cognition using ridge regression and 5-fold cross-validation. We will use a dataset from Human Connectome project, including the fMRI time series measured on 86 brain regions and total cognition metrics in 420 healthy unrelated individuals. We will share these codes with you, but I would like to go over the codes line by line to present you what each code does. Let's start. So the first part of the codes include the packages and functions that will allow us to visualize data and apply ridge regression and 5-fold cross-validation. And the second part of the code will allow us to visualize the data that we will use in the ridge regression to predict total cognition. So these lines of the code will allow us to load the data, which are the fMRI time series, and this part of the code will allow us to visualize the fMRI time series in the first subject. So when we run this code, we will obtain a figure like this, representing the fMRI time series for the first subject. So x-axis represents the time points and y-axis represents the fMRI signal intensity. Since we applied a global signal regression, we will have both positive and negative values for the fMRI time series. And here on this figure, each color represents different brain regions. That's why we are seeing multiple colors representing various brain regions for one subject. And then the following code will allow us to create functional connectivity matrices. So before we create functional connectivity matrix, we create an empty matrix representing, including the dimension of 86, 86, and 420, because we have 420 subjects and we have 86 to 86 metrics for each subject. And this loop will allow us to create functional connectivity matrices for 420 subjects. So when we go line by line, this line represents the codes to load the fMRI time series, and this line represents the codes to create functional connectivity matrices from 86 brain regions time series. So we are performing the Pearson correlation to each pair of brain regions to obtain functional connectivity matrix. And this line represents the codes to write down the functional connected matrices for each subject. So at the end of this loop, we will have a matrix with a dimension of 86, 86, and 420, representing the functional connected matrices for all subjects. Since I can't really present or visualize the functional connectivity matrices for all subjects, I would like to present the functional connectivity matrix for the first subject. So when we run this code, we will obtain the functional connectivity matrix for the first subjects. And when we run the code, we can see this functional connectivity matrix that each cell represents the correlation of the fMRI time series measured on region N and region P. As I mentioned in the previous slide, upper triangle and lower triangle of the matrix are identical and the diagonal is equal to 1. And this functional connectivity matrix is purely computed using this fMRI time series for subject 1. So here, each cell represents the correlation of the fMRI time series measured at each pair of brain regions. And the following codes will allow us to obtain each region's functional connectivity node strength. Before we obtain the functional connected node strength, we need to create a matrix with a diagonal of 0. So this is what we are doing here. Rather than using a diagonal equal to 1, we are putting 0 because we will take the sum of the columns. So that's why we need to create a matrix with a diagonal of 0. So what we do for the subject 1, we say let's put 0 for the diagonal and take the sum of the columns to the functional connected matrices. And when we take the sum of the columns of the functional connected matrix for the subject 1, we create functional connectivity node strength. And this part of the lines will allow us to create the figure representing the functional connectivity node strength for the subject 1. So when we run these codes, we will obtain this figure representing the node strength for 86 brain regions. So on the y-axis, we can see the number of the regions starting from 0, or we can say it is the first region until the region 86. And here, each color represents the node strength of this associated region. So when we take the sum of the columns of this matrix after we assign 0 to the diagonal, we can obtain this node strength for 86 brain regions. So this is how the data looks like. First, we have the fMRI time series. Then we can convert this fMRI time series as the functional connectivity matrix for all subjects separately. And then we can calculate the node strength for 86 brain regions for each subject separately. And what we can do afterwards, we can concatenate the node strengths for each subject to be able to apply a machine learning technique to predict an outcome. In our case, we will predict total cognition. So in the following section, we will represent how we can predict total cognition using functional connectivity node strength by applying ridge regression and five-fold cross-validation. So the first part of the code will allow us to load the data, including functional connectivity and total cognition. After we have the functional connectivity matrices, this part of the code will allow us to create the node strength for functional connectivity matrices for 420 individuals. So in this part, we are taking the functional connectivity one by one and putting 0 to the diagonal of the functional connectivity matrix, and then taking the sum of the columns to calculate the node strength for functional connectivity matrices. And then in this part of the code, we are removing the subjects with missing data, with missing total cognition, for example. And then in this part of the code, we are identifying the number of the subjects in the original data set, and we are creating the folds to be able to apply 5-fold cross-validation. And in this part of the code, we are creating a range of the hyperparameter. For example, since we will apply ridge regression, this is the range for the lambda parameter that I presented in the previous part of the presentation. And then before we start nested cross-validation, we need to create the empty matrices to be able to write the predicted cognition here and the beta estimates from the model. And here we can start for nested cross-validation. First, we are creating the training data sets for the input here and for the output here. These are the training and test data sets for the input and for the output separately. And then we can identify the number of the subjects in the training data set. And then we need to start for the inner loop. And here in this code, we are creating the folds for the inner loop. And then again, we are creating an empty matrix to write the correlation between predicted and observed cognition metrics for each hyperparameter separately. And here we are starting for the inner loop. In the inner loop, we are again creating training data sets for the input and validation data set for the input and output separately. And in the inner loop, we are again creating an empty matrix to write the predicted cognition for each hyperparameter value, because we will try different hyperparameter values. It means that for the ridge regression, we will use different values of lambda, and we need to write what is the predicted output for each lambda parameter. And in this part, we are testing different hyperparameter values, which is called alpha here, but in our case, it is lambda. We are trying different values of lambda, and we are predicting cognition using different values of lambda parameter. And at the end, we are calculating the correlation between the observed and predicted cognition to be able to identify which lambda is performing the best correlation. Means that the best correlation means the best performance accuracy in predicting cognition. And what we are doing this for each lambda parameter and each fold in the inner loop. Since we have 5 different folds, we need to create a mean or average correlation for each lambda separately, because we have 5 different validation data sets. And then we are finding the best hyperparameter that maximize the prediction accuracy. So these are the mean or average correlation for each lambda, and this line of the code will find the best hyperparameter that maximize the correlation. So this part, max HP functional connectivity, is the best hyperparameter that maximize the correlation in the training data set. After we find the best hyperparameter in the training data set, we can fit the model using this best hyperparameter and training data set, and then we can use this best model to predict cognition in an independent data sets that we were holding since the beginning. So test functional connectivity is the data sets we were holding since the beginning to test our best model. So this line will create the predicted cognition, and this line will create the beta estimates from the model. After we repeat this algorithm for each holdout test data sets, we can obtain the mean correlation or explained variance over 5 holdout test data sets. And these lines will allow us to visualize or compute the mean correlation and variance explained over 5 holdout test data sets. So when we run this code, we will obtain this results. For example, in our case, the mean correlation, this is the correlation between the predicted and observed cognition using functional connectivity node strengths. is equal to 0.20, and the variance explains is around 0.04. And this line of the code will allow us to plot the predicted cognition and observed cognition using this functional connectivity model. So this is the observed cognition, and this is the predicted cognition using functional connectivity node strengths. And here each dots represent two different values, one observed cognition and one predicted cognition. And we can create a line here. It is the linear model showing how well we can predict how well we could predict cognition using functional connectivity node strength. And here we can visualize the beta estimates or the future importance for each region. So these are the metrics that we obtained using 5 training data sets. And here you can see higher values representing, higher absolute values represents the best future importance, and lower towards 0 represents the regions less important in predicting cognition. We have the box plots for each brain regions, again because we used 5 different training data sets, since we used 5 cross-validation. So here are the results. If you have any questions, I gave our email address during the first part of the presentation. Please do not hesitate to contact us if you have any questions about the questions or about the data sets or about anything that you might think of about applying statistical approaches to vectorized connectomes. Thank you very much for listening.